Welcome to Fever Pattern, where every symptom has a microscopic source. In our first episode, we looked at the broad architecture of the bacterial cell envelope. We noted that gram-positive bacteria rely heavily on a dense, multi-layered structural armor called the Peptidoglycan wall, while gram-negatives have a thinner Peptidoglycan layer between the inner and the outer membrane. But to truly understand how antibiotics destroy a pathogen, we have to zoom past the surface and look at the molecular components of this mesh. Peptidoglycan is a polymer of two alternating amino sugars, N-acetylglucosamine and N-acetylmuramic acid. Bacteria can synthesize NAG easily, but the production of N-acetylmuramic acid is more complex. It's a multi-step enzymatic reaction catalyzed by the Muir enzymes, but we won't go into those details this time. After these reactions, we are left with an important precursor, UDP N-acetylmuramic acid pentapeptide. This pentapeptide tail is where the future crosslinks will be formed between the carbohydrates. While its sequence shows some variability, it frequently ends in a highly specific dipeptide of D-alanine. This will be important when we look at the antibiotic mechanisms and resistance. By the way, this precursor is also called Parkes nucleotide. After formation, Parkes nucleotide is anchored to the bacterial cell membrane via bactoprenyl phosphate. This enzymatic reaction is catalyzed by the MRAY protein. The resulting molecule is called undecaprenyl pyrophosphoryl N-acetylmuramic acid pentapeptide, or for short, Lipid I. Next, an N-acetylglucosamine molecule is added to Lipid I by the Muir G enzyme. This reaction creates Lipid II. Lipid II's pentapeptide tail is further modified. First, the bispecific enzyme MUIR-T GET-D amidates the second position D-glutamic acid to iso-D-glutamine. Then MUIR-N and MUIR-M add the dipeptide branch at position 3. Without these latter modifications, the PBPs can't form the crosslinks between the peptides. But there is a major geographic problem for the bacteria. These building blocks are manufactured deep inside the cytoplasm, but the wall itself must be assembled outside the plasma membrane. To bridge this gap, the bacteria deploys a specialized lipid carrier molecule called Lipid II Flipping. Think about it as a molecular ferry, which flips Lipid II across the plasma membrane. Once outside, our building block is finally ready for polymerization. A team of enzymes called penicillin-binding proteins, or PBPs, takes over. The first step is transglycosylation, where 1,4-glycosidic bonds are formed between the carbohydrates. Then during transpeptidation, other PBPs cross-link the interfacing mirapeptides to create peptide bonds, while the terminal D-alanine gets cleaved away. Repeat this a couple of times and our peptidoglycan is formed. And the best part? Antibiotics can interfere in various parts of peptidoglycan synthesis. More about that in episode 3. Don't forget to like and subscribe to the channel. And as always, thanks for watching and see you next time.